I'm very excited to introduce our speaker today. I'm Dan Eiserman. I'm the unit leader for the Wisconsin Cooperative Fishery Research here on campus. And I used to work for the Minnesota DNR before I came to Stevens Point, and I actually used to work with the person, Andy, that's here to talk to you today about some really interesting stuff. And so Andy had come to us in Minnesota after finishing his Ph.D. at the University of Wyoming, working with Frank Rahill and Wayne Hubert, looking at trout movements affiliated with irrigation use of some rivers, and he had a pretty extensive background in telemetry, and that really allowed us to do some really interesting things that we had not been doing with Minnesota. So he's been there now for
12 years?
Yeah, coming up on 12 years, I was going to say. And without further ado, I'll let him talk about the habitat selection of pelagic fish in Minnesota lakes.
All right. Thanks, Dan. Thanks also, Wes. And it's been a pleasure to come out here. I got to meet with a bunch of students this afternoon. It's always great to share this work. I guess I apologize first if you were hoping to hear one of my old supervisors, Pete Jacobson, was going to come and talk about some of the backdrop of where this work goes, of our conservation efforts associated with what we've been looking at through Cisco populations. So at the very end of the slide or the presentation, I'll try to wrap things up together with 3 or 4 of his slides to give a little bit of perspective of how we're using his work at this larger landscape scale to preserve some of these pelagic habitat zones. And certainly if an opportunity comes up again to have Pete come out, or if you see a chance to sit down and talk to him, he's got some really fascinating work. So as Dan mentioned, I've been with the state of Minnesota for about 12 years, and I was a stream trout person through my kind of first 10 or so years of schooling. Did a lot of fish movement work, and it tied into, at the time, to build off some of what Pete's work, looking at what was a perceived kind of declining of our Cisco populations in the open water. And one of the issues in Minnesota is we didn't really know what was going on in these offshore waters. We've done a really good job of monitoring the near shore. We used gillnets to sample for walleye and pike populations to track what their abundance are. But these fish that are out in the open water, we didn't know a whole lot about. So I was brought on to apply some of that, and so I grew up or did a lot of training in trout and been working with walleye, big surprise, and pike ever since then. So today's talk, I'm going to give a little bit of an overview of just how the Minnesota DNR works. I think I have 2 or 3 slides, how I fit into that mix, how we help answer questions, how we manage fisheries. And then I'll get into some of the definitions and tell a pretty interesting story. This is maybe the most complicated we'll get. This is a really interesting kind of habitat selection matrix. If fish like certain temperatures better than other temperatures, and they need oxygen. It's some of the really innovative work that we found that fish do need oxygen too. So to start things off, Minnesota's managed lakes. We manage almost 4,400 lakes across the state. One of our primary gears we use are gillnets. We have a long history of using them back into the 50s to assess those populations. It totals about 4 million acres of water. And we divide that up just recently within the last year. We've had 28 management. Right in the middle of the state here, we have one of our premier walleye fisheries that we've added a new management area. So we divide that up among 29 fisheries management areas. In a lot of states, they'll use external, or use funds, Dingell-Johnson funds to fund research. But within Minnesota, similar to Wisconsin, we have 10 research offices that maybe 10 years ago we had 26 staff, and I think we're down to about 16 full-time staff that are spread across 10 offices in the state. I've been right here in the center of the office my whole career there, again starting off with Dan. And our coverage throughout these positions, we used to have offices throughout the Arrowhead. When we've contracted from, I think, 16 offices down to 10, our spatial representations. So cover areas across the whole state, problems dealing with cormorant bioenergetics to Cisco up in the Arrowhead to fish movement in the lower Mississippi as well. So I'd like to try to keep this as informal as possible. If you have questions, certainly let me know as we're going through, or there's some additional terminology I kind of skip over. But to start off, just some definitions of what I'm talking about. Pelagic habitat, we often think of this word in the context of out in the ocean, but what we're thinking about of here is just these open waters. We think about the littoral zone, the depths of where plants grow down Minnesota lakes, anywhere between kind of 12 to 25 feet, depending on water clarity. that's where most of our fisheries or walleye and pike fisheries are concentrated. And what we're talking about is the flip of that. We usually set our nets up in these zones. This is a little study lake just to the west of Brainerd. And it's kind of a little awkward to look at, but a 3D model, and it's basically a cup. The lake is a pretty sharp break to go down, then it's 30 feet all around. And these little lines are the different assessment nets that we set out there. So when we think about habitat in these areas, it's not maybe the traditional kind of physical structures, if it would be rock beds or nearshore woody structure, but we'll be talking about temperature and dissolved oxygen throughout that thermocline. And then also we get some interesting work looking at light related to these movements. So these nearshore areas, we do a pretty good job of assessing, but what happens in this open water is a little bit different. So why are these habitat areas important? Well, they can have a unique species assemblage. We look at cisco, for example. They're found in about 650 of our lakes out there. And if we sample those nearshore areas, in some lakes, the highest density lake, I have a slide from some hydroacoustic surveys we've done, basically a fish finder counting all those marks. Highest densities population in the state, we've never caught one in our gill nets. But it's just a mismatch of where those fish are. It can also be an important component to understanding ecosystem function. We look at different life stages. We have big pike transition from these smaller kind of sizes of fish, well, to really put on a lot of growth and attain these larger trophy sizes. Going out and utilizing these resources, such as cisco in the open water, can be a really effective model. And in fact, we've seen with our trophy fisheries, both with walleye, pike, and muskie, that systems that have cisco are more likely to have those bigger fish. And associated with looking at those declines of the species around, looking at our standardized assessment. I have a few slides going through of what our traditional methods are, and then how we figure out or make kind of inference from what we can learn from those nearshore areas to what's going on in that open water community as well. And all of this in the back of what's happening with these changing habitat conditions. So I'll talk a little bit about climate change and what's going on. That's not what I do. We look at the implications from those best models that we might have. If water temperatures are going to warm, what does that mean relative to the fish habitat they're in? How might that affect our management strategies? So in back of that, looking at warming patterns and increased levels of eutrophication. And this is just adding nutrients. We take a look at, we've had a lot of good cleanup in the 70s and 80s, limiting nutrients going into systems. Some degree nutrients help fish grow. At the same time, in some of these clear water systems, what we'll see is what adding nutrients does to these systems relative to the oxygen below the thermocline in our lakes. So to give you a little bit of background on the 4 species that we're going to talk about, just to frame kind of our one cold water species in Minnesota, this is the most widely distributed of the fish that we have of cold water species, again found in I think 650 lakes. And the interesting part here is it's kind of found at the tailing edge, both in Minnesota and Wisconsin, of their range. And their optimal temperature is in this kind of 10 to 14 or right around kind of like temperatures less than 60 degrees. And they need oxygen levels above 4 milligrams per liter. And I wanted to reiterate, why are these important? I mean, they can offer people go out and angle for these actively. They're a good smoked fish. You can actually in Minnesota go out and gill net for these fish with a license as well. But the main reason that we're interested in these as sport fisheries managers is the potential for them to be forage for other fish. They have the caloric value. It's like eating a sausage versus lettuce for muskies and big pike out there. You can eat, it's basically a 4 to 1 for the same amount of grams to eat these big greasy fish out in the open water habitat. A cold water fish on the southern end of its range, if we start to see those ranges contract, it might affect our potential to grow those big fish in the same amount of time. So here's the dispersion. Again, we have those 4,300 lakes or so that were throughout the state. And here's where the lakes that are found, a lot up in the Arrowhead. Brainerd is right in here, but this transition zone where that species distribution range comes right here. Given some of the amounts of predicted climate change, we might be nervous that this is going to shift up and change some of the work. And a lot of Pete's work has looked at, well, how do we protect some of those watersheds where those fish might be? If we're going to increase warming levels, there's not a whole lot we can do about that at a local scale, but maybe we can preserve that oxygen below the thermocline. So there's always cold water below that mixed layer that these cold water fish can utilize. And some of the work that got Pete started on this 10 or 15 years ago was this decline. We have these legacy assessment here, and then our modern assessments where we're looking at a catch per unit effort. We set a standard net out. We can measure how many fish get in there across the full suite. And what we saw beginning in the 70s is this decline going down. And this is really what started some of Pete's work, and then subsequently when I was brought on, to be able to tease this apart a little bit. So looking at the mechanisms, is this real, and what might be happening associated with it? Again, there's that species range right at this southern edge of where they are. So contrast this with a walleye. We get up and give these talks. It's always, we take a look at, given the different levels of climate change, we're right in the heart, as you guys are, of the walleye distribution right here. Optimal temperatures in that 68 to 75 degrees Fahrenheit. It's pretty good to be a walleye, even with some given climate change scenarios. Will probably be pretty good as well. Now, depending on the productivity of the system, we might see some shifts. Maybe we can't grow them quite as fast or attain the same sizes, but we're always going to have walleye, and they're going to be doing pretty well. Oxygen levels similar to what we might expect for Cisco. Muskie, one of our a large cool water species, kind of just around the Great Lakes region is their distribution. Similar temperatures as walleye. Both muskie and northern pike can be able to do well in oxygen levels with a little bit lower. And contrast that with pike, again, right in their heart of the range to go up. And this is fairly unique. We'll be talking about in the Brainerd area right now. We're starting to go through some of we've had early snow on our ice. A lot of our shallow water lakes are starting to die from winter kill. What happens is we use up all the oxygen in the water, and we'll start seeing fish die. But pike are one of the species that generally does pretty well. They can tolerate really low oxygen levels, and in kind of the center part of these cool water range here, can tolerate the warmer, even under some of our more warming scenarios. So what is climate change going to mean in Minnesota? And again, we just use the data that comes out, but generally it's expected that we're going to have warmer weather in Minnesota. And what that means is the extent and the duration of summer stratification is going to increase. Other ways of looking at are the number of days that we have ice off in a lake, or the flip of that is how many ice-free days do we have, or the number of growing degree days. And you think of at the beginning, as soon as the ice goes off, lakes start accumulating heat. They do that until fall turnover. We start putting ice on when it's fixed again. In that time, we can accumulate heat. And if we're looking at the cold water habitat in a system, well, the longer that we have those open water time periods, the more heat that they can accumulate. What does that mean? Well, we're going to start, we always have, or most deep lakes have a cold water refuge, but if we use up that oxygen in the bottom, these fish are going to be pinched in these center layers, especially our cold water species. So what does this mean? Well, if we have warmer weather in some of the air temperatures, depending on the lakes, we're going to have warmer water temperatures. We're going to increase the duration, extent of that summer stratification again. We've done some modeling that we're going to see warm species expand. Currently, we look at like bass populations. The northern side of their range is kind of the northern side of Minnesota, but densities and numbers are going to be able to shift to the north. At the same time, we'll see cooler water species kind of following those same shifts moving northwards, and we'll see cold water species decline. In some cases, we thought that we might see declines of upward of 40 to 60% declines in these fish populations. This was a picture taken a few years ago. I think in 2008, we had a pretty hard summer, a pretty long, we accumulated a lot of heat. We didn't have a lot of wind, and these are the Cisco dead up on the shoreline. So just to give you an idea, this is a little bit dated, but I think it helps frame some of the ideas as we prepare for some of the pending. There's always a lot of uncertainty around these models, but when we take a look at what happened with Minnesota by the end of this century with what winter conditions look like, it might be more like present day here, which I've been in Minnesota, originally from upstate New York, and didn't realize how cold it can be right here in the center of the state with all these winds coming down here. But I would welcome Wisconsin winters. But we contrast that with some of the predictions. This is the most extreme prediction of what it might look like summer-like conditions. And again, it's a little bit more kind of where I grew up. We didn't have these hot, humid days that we do get some of those in Minnesota, but I'm sure down here in Kansas it's quite a bit worse there. And you think about that species distribution of those cold water species coming through like this, not a whole lot that we're going to have for the overlap of the temperatures in that. So I like to frame a little bit of how we look at these temperature and oxygen. It has to do with how lakes stratify and turn over. And just as a little bit of a reminder back to the days that we took limnology, or if we didn't take limnology, of how lakes stratify throughout and turn over through the different times of the year. And as we take a look at during the winter, water's kind of interesting in so much as it's most dense at 4 degrees. So we usually think of warm temperatures, if it's air or any material, rising. But water is densest at 4 degrees. So during the winter, the water is coldest at the ice-water interface at 0 degrees. Then water is densest at 4 degrees at the bottom. So we actually have a temperature gradient during the winter. Then what happens in the spring, the ice melts off. We start warming up that 0 degree water at the ice-water interface till it hits 4 degrees. Everything is the same density here. We get complete turnover in the spring. Then what happens in a lot of our lakes that, depending on how long they are and how deep they are, wind can mix the layers. If a lake is relatively shallow, it can usually mix down all the way to the bottom. In a lot of our lakes, in between 25 and 30 feet in Minnesota, depending on how wide they are, the fetch that might go through determines how much we can mix this top layer. And what we'll see is this epilimnion is this kind of this upper water that can mix throughout the summer, again, the top 20 or so feet. Then in the metalimnion, or where the thermocline is where we see this rapid drop in temperature. And then the hypolimnion is that area below, that once we seal that off, we don't get to replenish any of it. So we have a fixed amount of oxygen in that lower, below the thermocline that we can't mix up and get any more. And I'll come back to that process of how that works. Then what happens in the fall, what we'll do is we start losing heat from this top layers. This 22 turns into 20. All these temperatures start dropping until everything hits 4 degrees. Then we get complete turnover in the fall. Then we get ice on, and that same process sets up again. So most of the work I'm going to be talking about is how fish respond in these 3 zones right here. So, talking about using up oxygen. Well, when I did all my stream work, I didn't think a whole lot about this, but it turns out we can run out of oxygen in some of our lakes. And how this happens is through this eutrophication, or adding the nutrients. And I put together this diagram that I think helps frame it fairly well. Much like a garden, if you go out and fertilize your garden, you'll have stuff grow. In this case, we'll have an increase in photosynthesis and algae bloom. Get this big bloom. Then what happens, and we get that thermocline, the metalimnion right here, it goes down below the thermocline. The algae dies, oxygen-demanding bacteria decompose that detritus to come through, and it uses up all that oxygen. So depending on how many nutrients and how big that reserve of cold oxygenated water is, what we'll see is these hypoxic conditions come up, come up, come up, and in some cases, all the way to the level where we mix all the way up to the epilimnion. So while there could be good cold water in our systems for our cold water species, they might not have enough oxygen. That's driven by how many nutrients we put in those systems. So I always like having some animations of fish moving around, some kind of interesting conceptual models. So during the spring, we have fish. They can swim all over the water column. This is a Cisco. They're really happy. But then what happens? We start warming things up. It's too warm in the upper water. We start using up that oxygen. What happens to our fish? Well, they're truncated. They're funneled down in this area, probably still pretty happy for the most part. Well, we're getting a little bit later in the summer. We're accumulating more and more degree days, using up more of that oxygen, truncated even more. Well, the fish are starting to wonder what's going on. And then those extreme conditions, what happens? There's nowhere for the fish to go, and they die. So then what happens after that during the fall? They can move back throughout the water column. So really interesting idea is how do we do some research, and why is that important for research? So the pelagic habitat, this is that open areas where these fish are going to respond through. We're looking at the depth on the y-axis, just the date through throughout the summer. And what I did is I just recorded the depth, or interpolated the depth at what there was 20 degrees C. So this is kind of that upper level for where Cisco want to avoid. And this is that 4 milligrams per liter, the depth of where it was. So these 2 different habitat variables that we can see that they're getting squeezed and squeezed and squeezed down. And we can also look at light, which I'm not going to talk about today, but another important variable that might drive how much they move within these habitat zones. So how do we go about assessing that type of habitat that's out there? Well, we can go out and just directly measure it. We have these probes that we can go out once a week, and temperature accumulates in a pretty linear manner as you go through. So once a week does a pretty good job. Lower our little probe in meter increments throughout the water column, and we can interpolate that to see where it might be. It works good if you're close by, But the other ways, like we got some study sites across the state that we'll put together these I call temperature chains or oxygen chains. And we'll put a buoy either on the surface or 2 or 3 feet below the surface. And then we'll stagger these temperature loggers, and they'll record the temperature of the water every 15 minutes, every half hour, whatever we program it to. And we also, in some of our more recent work, have put these oxygen sensors on here as well too. So we don't have to drive 3 hours to the other end of the state to do these profiles. We get nice continuous data throughout the day, across weeks, across years to go through. So now we've got these 2 different pieces. We've assessed some habitat. We've got this nice, again, depth as they go through. What are we hoping? That our fish can sneak through these little areas. And what we'll see is those extreme conditions that I talked about, as you can actually see, well, if we're continuing to accumulate heat, still increasing growing degree days, accumulating hypoxic water, when those 2 lines cross, Pete had an interesting paper a few years ago that came out that we verified here is, in fact, when there's no habitat, the fish do die. So a little bit of background of how do we know what's going on with the fish. We've not too innovative techniques, just going out there and measuring the temperature and oxygen, but assessing what goes on with the fish community is a little bit different. And I'll talk generally about the 4 different species we have here, but for the most part our standard gear are the standard gillnets sampled for walleye and pike. Muskie are quite a bit larger than usually get stuck in them, and cisco are usually in some different habitats. But some ways that we might look at them would be our standard nets, suspended nets, vertical gillnets, trawling, hydroacoustics, and acoustic tags. And we'll go into each of those just to kind of give you guys an idea of what they are. So our standard bottom net, gillnet set, and this is kind of the standard technique that we use in all of our lakes across the state, are focused on this near shore area. Again, that near shore kind of littoral zone, less than 25 feet. Most of our nets fish in kind of 15 to 30 feet of where they are, sampling the bottom 6 feet. Got 5 different meshes as we go through, and it might intersect some of our fish populations that are in those zones, but it might miss quite a few that are out there as well. So primarily the sport fish that we manage are in this zone, but sometimes when they get really big, they might kind of trade habitats as well too. Well, we can take that same net and just suspend it. We'll go out with our little temperature oxygen meter and figure out where that perfect temperature is, or at the thermocline, and say here's where we might think fish might be. We can put buoys along the top and just suspend it there, and it does a really good job at catching fish if they're there in those zones. Or another tool that we use are these vertical panel nets, and we've actually worked with some of the Wisconsin DNR folks as coming up with similar methods to sample these offshore fish. But what these are just gangs that are 10 feet wide of different meshes, and we'll roll them from the top all the way to the bottom to go through. And they're metered throughout the water column, so when we roll them back up, we can tell how deep the fish were at the different depths in the water column, where they came from. So here's a picture of our boat right here, just kind of unrolling this big spindle that goes all the way down to the bottom. When we pull it back up, we see these fish, and it'll say, all right, this fish was 10 feet from the bottom, 15 feet from the bottom. It'll keep track of them. And then we can match that up against the temperatures of where those fish were. We have a few different sizes of mesh to catch those fish, and we use it to complement hydroacoustic surveys. And again, this is your fancy fish finder that records those little arcs. I've got a slide coming up to show it, but we'll use to divide up those arcs based off the assemblages that we see in these different gangs of nets. We can do trawling. This is done in some of our large lakes, but generally we don't do a whole lot of it in Minnesota. And then hydroacoustics, which is your fish finder hooked up to your GPS and a computer. And what it does is it just pings back and forth on transects in the lake. Depending on the fish size, it'll kick back different sizes of marks, which we can use equations to calculate how big they are. We can do this at both the day and the night. Here's a small lake that we just have some transects. This was a program that I think I was handed fairly early on in my career, and I quickly realized that I was not an expert in this, and we have subsequently hired 2 limnologists who have taken our program, reworked our Lake Superior. We use this tool to actually set our quotas in Lake Superior for our cisco catch now, and for inland cisco populations as well. But the transducer on the back of your boat is generally about the size of this little clicker. This guy right here is about, 20 inches round, and it weighs 40 pounds. What we do is there's an arm here that flips up, and we can lower it down in the water. Little box runs it, and there's a computer that records it. Analyze the Great Lakes standard operating procedure. And we have our own kind of operating procedure now too. And here is the highest density lake of Cisco that we have in the state of Minnesota. This is 10 Mile Lake, south of Walker in the Hackensack area. These are marks, all individual little fish, 10 meters, 20 meters, 30 meters, 40 meters, 50 meters, 60 meters down. And how bright blue they are, or how light they are, is the size. You can see there's a bunch of schooled fish here, but you can see there's no fish in this upper warmer water. There's kind of a layer here, and we can go through, use some tools to count how those marks are, and depending on how blue they were is equivalent to that target strength. And this is a negative scale here, so these are the smaller fish, these fish that have a reflectance of 50 or around negative or 1 to 2 inches, and so on. So we can use this equation both by depth, and then what we do is we have those vertical gillnets, and we can say, all right, how many fish, we have those different meshes that would cover the different sizes we might encounter, that we'd say, all right, well, of the fish that were between 10 and 21 inches and at this depth, our nets would say that many of them are Cisco, but there is some overlap. So what you'd hope is the species that you're really interested in looking at is a pretty clean mark right here. There's not other fish in here. But what we would say is just of the marks here, 80% of them would be Cisco, 10% of them would be walleye. So a pretty interesting, innovative way that we're still in the process of figuring out how to integrate into our monitoring programs, but utilizing a tool that a lot of our anglers use already to use in a scientific way. And then finally moving into the types of work that I use, and this is acoustic tags, and what we'll do is we surgically implant fish with these little tags, and it's the same technology that you'd have in a transducer, but what happens is this tag pings out the information. We have these submerged listening devices, so when a depth finder works, is it sends sound down and it reflects back up off the bottom and it records that time, and that's related to the depth of what it is. Well, what this does is send out the information in encoded ping, and it's just heard on this device. So our fish will swim around pinging out information, and there can be sensors in these tags that'll tell us the pressure, which is equivalent to the depth, and the temperature of where those fish are. So I've been doing this for 8 years now. We've tagged Cisco, walleye, muskie, and northern pike. And the real interesting kind of angle here is we'll get continual movement. We'll get a location every 1 to 2 minutes on fish. We have that high resolution temperature and oxygen map. So if we know the depth of where a fish is, I can go back to that habitat map and say, well, here's the temperatures and oxygens that they're in. And additionally, we can have a tag that's inside that gives us the internal temperature of the fish. This is just one of the Ciscos we sacrificed to make sure that the tag was sitting right. And in fact, they do really well. The first year Dan and I were out there, we tagged 8 fish, or maybe it was 10 fish. Nobody expected any of the fish to survive. For the full sampling window, all 10 fish lived throughout the summer. And we have an incredible, probably one of the best data sets we have to date on Cisco through a season. So, kind of reiterating here, we have these listening devices around. Depending on how loud we have that fish pinging or that tag pinging, we can determine how, where, and how we want to arrange our arrays of hydrophones. We can compare between species and lakes. So we'll take a look at some data now. So we've talked about the habitat, we've talked about how we're going to assess fish. We've looked at these graphs before, looking at how deep the water is, looking throughout the summer. Here's that hypoxic layer at 4 milligrams per liter, and here is the depth where that 20 degree C is. I'm going to come to the secondary Y in a second, but just to start, we're going to bring in a blue series of line. There's going to be 40,000 data points that zoom through right here, and this is where that fish was throughout the water column throughout that time period. So during the beginning of the year, fish are moving up and down, up and down. This really hot day, our fish stopped moving. This is just one fish that we looked at, but all the fish across all species stopped moving for about this 2-week time period. And this is where they're starting to get squeezed, not moving a whole lot. But you look at that fall turnover, following that hypoxic layer down, utilizing that whole water column again. So this is just using the depth. We also have internal temperature on these fish. We're going to look at that secondary y-axis here, looking at different water temperatures. That same time period, we've hit, this will come back to an important kind of, it's about 15 degrees as we look here. Fish are moving around, they're probably pretty happy. Now they're starting to be stressed. They're going through in the warmest water that has adequate oxygen till turnover. They're just kind of holding their own in this window right here, down into the cool water. So really cool data on individual fish. And again, lots and lots of data here. I think there's 80,000 data points between temperature and oxygen on one fish. Been doing this for 8 years. I have 17.5 million records of individual depths and temperatures of fish. So much data we don't even know what to do with. Usually we're worried about sample size. When you run these things in models, the traditional models, all variables usually come out significant because there's so many data points in there. So our research sites, and this is some collaborative work I've done with our now both retired walleye and muskie biologists. But in Pelagic Lake for 3 years, we looked at cisco, walleye, and northern pike. Little Wabanas up near Grand Rapids, looked at pike and cisco. Shingabees up near Walker for northern pike. And then Elk Lake is up in Itasca State Park. We had some collaborators with Muskies Inc. who were even glad to help fund us some work looking at cisco, an important forage species for the target species that they like to chase around. And also looked at northern pike and walleye. So what are the questions? We like to see fish moving around, ecologically really cool, but why does that matter to management biologists? Why does that matter to research scientists? Well, we can take a look at the species interaction, the habitat overlap, looking at the timing and the magnitude. We have a cold water forage species and a cool water predator species. Well, how often do they intersect? Do they intersect? And then we can look at this habitat, pelagic habitat preference. Well, are there certain depths, temperatures, and oxygens? And that becomes really important when we come back to our assessment techniques of gillnets. So what we can do is, based off those habitat variables, is model the species depth, which correlates with using temperature and oxygen. And then we know where our nets sit. And we can say, well, do our nets fish in areas? For a fish to get stuck in a net, it has to move as a passive gear. So if we set the net in the area that's in a temperature oxygen where fish aren't, they can never be caught. But if we align those up with those habitat ideas, now show us how we take the model to where those depths are. And then potentially we can do this habitat stratified sampling. What we've done in the past is we use these same fixed sites, depths, locations to go through, but depending on our questions, we might target a net, or we could stratify our sampling effort among those different depths of the probability distribution of where fish might be throughout the water column. So here's Elk Lake, a beautiful lake up in Itasca State Park, the headwaters to the Mississippi. Wilderness Drive kind of comes through here. If you're ever through here, this is maybe the unofficial headwaters to the Mississippi. It outflows into Itasca Lake right here. You can see we've got the different little black dots are the different hydrophones. There's 10 of them throughout the water column. We've had a temperature line and an oxygen line out here in the deepest part. We also use light, and I won't talk about it here, but we just have these little light sensors throughout the water column that throughout the day we can take a look at water clarity and how deep water gets at different times of the day. And depending on the fish's eye, how they respond to different light levels, will determine how active they are at those different times of the day. So we'll take that habitat data, combine it into half-meter increments. The deepest spot over here is about 100 feet, so 60 zones throughout the water column. So then what can we do with that data? This is about as high-resolution habitat data as we'll get, looking at the temperature. This is Pelagic Lake again, looking at the 12 meters down throughout 4 different years. And what we'll see is just this continuous between 30 degrees down to 0. And we can see during the first year, we're getting warmer through the heat of the summer. Then we see turnover, cold water during the winter. Then the next summer, we see stratification set up. And again, we can see this thermocline right around 5-6 meters in Pelagic Lake. So we have 4 years of data. We can start comparing between years. We can see, well, if you look at the accumulation of growing degree days, the summer of 2011 was the warmest to go through. So when we have fish out here pinging at different depths, we can do that same overlay and see, well, what depths were they at. We can do the same thing for oxygen as well. It's a little bit different. As I talked about, the lakes turn over twice a year. And we can get hypoxic conditions in the winter as well. And what we'll see going into 2010, this is the summer hypoxic layer, turnover in the fall, the winter hypoxic layer coming up, spring turnover right here, hypoxic layer in the following summer. Is a really high oxygen event, and it was consistent through 2 or 3 surveys that we had there, but really high levels. But then we can just contrast, well, how do fish respond to both the temperature and the oxygen that we have there? So to take a look at the species interaction, again, looking at 4 different species, the predators being walleye, northern pike, and muskie, prey being cisco. We can look at the timing, the magnitude of that overlap. So Elk Lake, again up in Itasca State Park, kind of a mid-level productivity. It turns over twice a year. The deepest spot down here, about 100 feet deep. The data set I'm talking about here is just for kind of the 2011 to 2012 year. Had 5 walleye, 4 northern pike, 8 muskie, and 6 cisco. To complement that, temperature, oxygen, and light. So how do we start pairing that data together? There's 4 different tiers we can look at. We can look at the lake habitat, the local weather, the solar lunar trends, and then there's this fish data, which we're going to concentrate for this talk. And we're going to focus on these annual trends of the species just to kind of give you an idea of seasonally how the depths might change and that oxygen preference. And then tie it together during the summer. We set our standard nets between June and August to go through. So where are the fish when we're trying to assess them? So here's some data from Elk Lake. Again, you can see a little bit deeper, down to 30 feet to go throughout the summer. We've got that 20-degree mark, and they also put a 24-degree mark to go through. So what we'll do is we're going to focus on this kind of summer time period where we do our assessment nets, but I combined the data from these 4 different species over the full time period. And what I'd really like us to pay attention to is, especially if we're anglers, we like knowing where fish are. Halfway between these 2 dates, no, actually 210 is when ice set up. Halfway between these 2 dates are when ice went out. So we're going to watch kind of about what happens to fish movement when they hit either of those 2 things. You think lake habitat probably changes pretty substantially when ice gets on it or ice goes out. So what I did is I just took kind of an average daily depth for each of those 4 species, combined them as we went through. So it wasn't a very good year for cisco in 2011 as we went through. We tagged, 6 fish, and by the time they hit that summer window, all fish were at the bottom. So if I maybe would have continued this, we would have saw the average go down to the bottom of the lake, which shows us that they're dead, which is, and they continue to ping. So throughout the rest of the study duration, as long as those tags were out there, I could verify that the settling rate in the mud of Elk Lake was that much a month or something. But certainly down in these deeper waters before that hypoxic layer set up, we contrast that with what's going on with muskie. And these are pretty interesting. We'll see they're getting through the season deeper and deeper and deeper, following turnover, down into pretty deep water. The day ice goes in, and this is on average, but these fish were right under the surface, within the top kind of 10 feet of the water, till the exact day that ice goes out. Then they go down deep on average. These are fish coming up to spawn in the spring. We can look at, as individuals, how often they do it, how often they go back and forth between those deep and shallow water environments. We can also contrast that against northern pike that spawn 2 or 3 weeks in front of when muskie do. So we can see pike coming into the white line, kind of up and down, maybe a little bit deeper, but these neat cyclic trends going through. And during the winter, at least in the Z, the depth is when they're moving the most throughout the water column, coming up to spawn in those shallower waters a little bit before our muskie do. And finally, taking a look at walleye. And as a walleye angler, what do you do in the fall? You go out in deep water, Lindy rig for fish, and why does that work? Well, on average, it looks like our fish are out in that deep water, following that turnover to go through, slowly coming up about the same time in those spawning areas to target as northern pike do. Pretty interesting cyclical patterns here associated with walleye and northern pike as well, but another story. So the other thing we can do is look at the variance. So how much are those fish moving up and down? What we'll do is we'll look at the secondary y-axis over here, which the closer you are to the list line, the less you're moving, the higher up, the more you're moving. So right before a cisco die, it's not surprising, they don't move a whole lot. But we contrast that with what happened with muskie during the summer, certainly that top layer after turnover. This is that ice period right underneath the surface coming in to spawn in the spring. This, I think, is the most interesting one. We don't know a whole lot about what northern pike do in the winter, but they're moving up and down throughout the water column quite a bit, as well are walleye. So some really cool data we can combine to take a look at just those locations of where fish are. So the first step of looking at this, without looking at habitat selection, well, we have these depths of where fish are. We've got these good bathymetric models or good maps of the lake at the different depths, and we can sum that up by area. So kind of in the dark outline, we can see how much habitat is in these shallower waters versus how much is in the deeper waters. And we can just plot the percent of the fish observations at each of these depths. So look at the use versus how much is available by depth. Here's muskie, you can see right about 3.5 meters down is where that peak is. We can contrast that among our pike, which are maybe a little bit on the cooler side there, but fairly similar to muskie, and cisco, just absent from that top water, which is fairly warm, absent, all these species are absent from these low hypoxic waters. And then looking at walleye, which are a little bit more in those shallower waters as well. So kind of the base, we have some areas by depth that we know they are, and then we know how much that volume is out there. But we can do a little bit better. And I just wanted to point this out because it's really cool data to start with, but we can look at these diel trends as well. And this has to do with what's driving by light. So we're looking at 4 different species in Elk Lake. I'm just going to start with cisco, looking at, what are we looking at, about 8 days or 10 days, but you can see what happens, and I have a slide coming up next showing when it gets light, at certain light levels, cisco start moving up and down. At night, they don't move. They start moving during the day, night, night, going through, which is really pretty interesting. But what we can then do is contrast this looking at the overlap with a pike. We don't have quite as much data on these, but a little bit higher. Again, our cool water species, our cold water species, our first cool water species being the pike to come through. We can look at this overlap time period of when they might come through. Contrast that with walleye. So again, remember this is cisco move during the day, walleye move at night, going up and down. My guess is during the day they're going up. Elk is a pretty clear lake. They're sticking their heads in the vegetation, waiting to not get eaten by this guy who's also up in that same area. So we can see when these muskie are super active during the daylight hours. And just to zoom in, because we like thinking of if you're a muskie angler, they're really hard to catch. Well, where are they? Before I get there, we can talk a little bit more about this overlap time period. Elk Lake has been a collaborative research site for us that we've had a lot of our staff putting time in. Brian Herwig is one of our scientists up at the Bemidji office and did some stable isotopes, looking at the ratios of nitrogen and carbon. And what they can do is look at the different species that might be out there to develop the signatures, then go out, take some cores of our muskies, and they can see that up to 50% of muskie's diet right here is made up by these nice greasy cisco to go through, followed up by white suckers and other things. So to take a look at these relatively small areas where they might overlap are actually making up 50% of their diet. And this is averaged over a year. There's certain times of the year when you see them going deep during turnover. That's probably a great time for muskies to feed on cisco. So just a little bit of a teaser to look at light and how it works. We're looking at the secondary Y here, just the amount of light. At night it's dark, at peak, at high noon it's the highest light intensity. This is real data that we monitor out in the field, relatively cloudy days, a bright shiny day. And just to kind of show the overlap of the depths of where this 42-inch female was. So during the night, right up at the surface. During the day, coming down, going back and forth. And we can look at cisco do it when you saw that other slide to come through of that activity of, well, do they school up during the day to avoid these predations? But unquestionably, we're seeing these large predators go out and utilize these deeper water habitats. And at night, they're up at the surface. So pretty neat understanding of what these fish are doing. So how do we put this kind of unique fish data, makes really great graphics, but put it together in a modeling framework that we can use to interpolate to some of our profiles? And this was some data done in Wisconsin back in the mid-'80s, looking at cisco lakes and using vertical gill nets. They'd set nets out there. They could see the depths and temperatures of where they are. And we're just looking at a relative selection curve as a way to think of it. So we're looking at temperatures across this axis, oxygen levels. And what they did is, this is kind of the earlier work that if you look at a cold water species, right around 12 degrees is that optimal temperature, plus or minus 2 degrees on either side. And what they found is that in liking oxygen levels at greater than 4 milligrams per liter. So they were able to generate this model and say, all right, well, here's where cisco should be from our net data. And I'm like, well, that's really cool. I should be able to do that. I've been following cisco for 8 years. I've got 10 million data points or something. How come I can't do this? So I tried to do it. And what I saw is when we're looking at real world kind of habitat, we're looking at temperature, our coldest temperatures, our warmest temperatures, our lowest oxygens, and our highest oxygens. I can go out and do a profile. And what I do is I don't get all combinations of this habitat space. So here's the first 10 days of July and the combinations we have, the second 10 days of July, all the way as we go, June, all the way through the end of August. So you can see we're getting slowly warmer. We always have this hypoxic water down in here. So at the end of that 10 block series that takes us from June through the end of August, this is the only area, that habitat, that my fish could be in. So to test that, we looked at walleye over 2 lakes, this temperature and depth, a little over 200,000 records, a mix of males and females. And what we could do is then match the depth and temperature to the oxygen. And you got to weight by the number of observations you have. There's some things you need to do to match up kind of our data sets. And I always like looking at that raw data first. There's lots and lots of data pieces. If we're thinking 100,000 records for each of the 2 years, I can just kind of standardize by the number of fish and locations. And what we'll do is go for 2010, 2011, that temperature starting in June to August. And what we can see is this green line is just the median temperature by day, the 25th percentile, the 75th percentile. So just that inner half of the data of where fish are. And there's some variability, but right around 24 degrees, right around 24 degrees is where they end up. Not very often this cool water habitat, but there's not a whole lot of this cold water habitat in the anyway. Can do the same thing for oxygen. And this was really surprising when we plotted this out, because when we go out and set a gillnet, we try to identify areas that have oxygen levels greater than 3 milligrams per liter, or 3 parts per million. But what we're seeing is these fish are hardly ever in oxygen levels that are below kind of 9 milligrams per liter in both years. So this is just looking at the raw fish data, again, that inner quartile of where they are. So what we can do is use this data. Here again is Pelagic Lake with our 6 different standard gillnet sets. We know that from just the habitat use, not what's available, we know that they're right around temperatures on average that are 24 degrees and 9 milligrams per liter, similar in the second year. And we can go out and do a profile. What we're looking at the depth throughout the water column. Temperature is the green, so here's the thermocline at about 6 meters. We have warmer temperatures, about 30 degrees down to 20 degrees down to 10 degrees. Oxygen levels, we get that metaluminum max here. Maybe we're around 9 parts, 10 milligrams per liter, then drop off to 0. And I can say, all right, well, I have my nets and I know the depths where temperatures are and oxygens, and I know where fish like to be, and I'll say, all right, my net's 6 feet tall, and I can say, well, these habitat areas are all too warm for walleye to be. So some of our nets might not do very well at sampling the walleye based off temperature. I can do the flip on oxygen, and I know that fish like being in better than 9 parts per milligram and plot that out, and these areas are all going to be hypoxic with that 6 foot. So these nets where these optimal zones are a much truncated part of the lake that we might look into. So that kind of helps us frame maybe a little bit difference from the depth and spatial distribution to kind of this habitat focused of where fish might be. So going back to we were just looking at where fish were, now we can also look at where fish weren't. So using that continual data, I added an axis here that's going to look at the relative frequency within each of those time blocks of where fish were. So here's where they could have been, and there's where they were. So very much avoiding these cooler temperatures and lower oxygen zones. And we can go through, we're warming, so we're getting more habitat up in this warmer zone. These are just where the fish are found throughout the water column. We start to see some warm water with not so good oxygen. They're staying up in this more oxygenated water, and there is some of this temperature up above 25 that they're avoiding, down in that 24 degree temperatures. Continue that throughout. We're starting to lose some heat here. This is probably the best spot for a fish to be right now, right at that 24 degrees in turnover. Then what we can do is combine all that together. We see these two plots. We can do that for 2011 and then the previous year as well. I think I combined it all. So overall, I can't create that really neat 3D graph using this technique, but this is just the observed data. So before we get that, the next step, we can look at these basic habitat selection. And we're borrowing a lot of this from the wildlife staff and their analyses. There's some really good statistical packages out there. And if we remember back to the statistics, we can say, well, there's an amount that is out there, and there's a certain amount that fish use. So we have to have our strata. We have different temperature bins, half a degree increments, and I can tell how much habitat's available. And then from that telemetry data, I know how much is used. Then we can look at the similarity between individuals, and then the key here is using these selection ratios to look at the habitat selection. So if you have a habitat selection ratio of 1, that would be an equal probability for if it's negative or below 1, they would be selecting against it. Above 1 would be selecting for it. So this is using the areas where fish weren't. So we can look through June through August, the 10 different strata. You shouldn't do this, but we'll combine it just for simplicity to take a look at where they are. And basically, the red areas are the spots that are avoided. The yellow are kind of neutral, and the green are the really good areas. And kind of the summary is we see this 20 to 23 degrees. This is the same work that they did in the 70s and 80s from the nets. That's really the areas they like to be. And we start to see this late season avoidance of these warmer water temperatures. We can also do this for oxygen. We see this seasonal trend going through that they're avoiding kind of, or they're starting to go in more hypoxic waters, probably avoiding those warmer temperatures as they go through, looking at that selection around 8 milligrams. So in summary, we have observed locations. This is just where they were, 20 to 23 degrees. Oxygen better than 9 parts. We can use this resource selection, which factors in where they weren't as well. Fairly similar, but we can really start to see this avoidance of temperatures greater than 25 and less than 7 milligrams per liter. So how do we fill in the rest of that graph? This is something that I graduated from school in 2006, and certainly always been in the fisheries field, but this is something that ecologists, wildlife staff have used for a while. These are these species distribution models. They can get multiple points on fish to go through, also called niche models or resource selection functions. What they'll do is they'll use a series of environmental data to overlay together on top of some locations of where fish are. You can combine these to model these predictions. And they use these fairly intricate models, or these mixed models, to account for the different year effects, lake effects, individual effects, differences in sample size. And this is something I didn't learn about in grad school, so I had to try to teach myself, this was probably 3 or 4 years ago, to sit down, and I was really scratching my head, not making any progress. It's fairly new. I don't know if it's in the textbooks yet. So looking through the literature, you try to find somebody you went to school with or you can rely on to help you out. And I came across a paper looking at plant responses in Cape Town, down in Africa. And if you notice this last name right here, we can reflect back, and there's a picture of us about 35 years ago. So lo and behold, my little sister, I sent her a data set that I've been working on for, we'll just say 6 months. She turned around in 4 hours and came up with a model that looks like this. So what we're looking at here is just, and the way that she uses these tools are to look at plant communities. And plants don't move very often, but their distributions are tied to elevation, water patterns, temperatures as you go through. She now works for the National Park Service as a biometrician, and she's modeling tortoise habitat. So she's like, I need to figure this out, take my app, apply it from the plant world into some real world plant stuff, or into animals. And she's like, I can figure this out for you. So taking the model to a whole other level, which is really pretty interesting. And what I'll go through this quickly. We have these warm temperatures, and this is for walleye, and we have low oxygen to high oxygen. And just want to zoom through kind of the utility. Let me get my mouse. So here's this model, and what makes this so interesting is it matches up with what we might think. So if we look at this front panel right here, dissolved oxygens at the highest level, temperatures at the warmest right here, so we're going to look at this plane right here. From all our other information, we kind of think it should be around 24 degrees is that max, and in fact right there it is. And we can look at this trade-off at different temperatures and different oxygens. So taking it one level further with real data to fill in kind of these gaps of where it is. So how do we use this really cool habitat selectivity model? How does that apply to what the Minnesota DNR does? So standard gillnet sets. This is Gull Lake. It's a lake just north of the Brainerd area, about 10,000 acres. We set 10 or 15 gillnets out there. To take a step back, generally we set nets between June 1st and August 31st on a 1 to 20 year cycle. We have 5 50-foot panels of multi-filament mesh from 3/4 of an inch up to 2 inches. We set at fixed sites, always come back to these locations on the same days of the year or week. The number of nets are lake and size dependent, generally set up to target walleye and northern pike. So, what do those nets represent? Like I said, the first thing we can do is look at the depths of where they set. We have some maps. Anglers now have these commercially available software. This is made by Humminbird, Contour Elite, that you can buy. Usually put it in your GPS so you can look at the different depth contours, but you can get computer versions. We can go in there and create these hypsographic curves. And what we're looking at is just the area. Here's 10,000 acres of the different lake depths. So we have 10,000 acres of 0 or 1 foot depth as we go all the way out to the deepest point here at 100 feet. So I have an idea of the depths of what is in the lake. We can do this for every lake. I can go out there and I know where our nets sit at these fixed locations. I can create maps using my GPS and depth finder on the boat. I can combine on the secondary axis looking at the acre feet of gillnet. We have a gillnet that's 250 feet long, made up of 5 panels of 50-foot mesh, 6 feet high, and I have 15 of them. Multiply them through and I can then look at the depths of where they fish. So now I know the depths of where they fish. How do we tie that to that oxygen and temperature gradient? So as part of every survey, we do a temperature and oxygen profile where we're looking at dissolved oxygen at the surface all the way down to the bottom. This is real data from Gull Lake. Temperature, here's that thermocline dropping to the cooler water. So to overlay this on our model, what we have to do is first interpolate this into standard half-meter bins to go through from temperature all the way to the bottom, and the same with oxygen. Then we can take that nice 3D model that we had, squish it down flat. We're looking at dissolved oxygen here from 0 to 10 or 12 parts, temperature from 0 to 25. Here's that 0 plane, so this is kind of a neutral selection. And again, we don't have these conditions, but when I have that standardized profile, I can say, all right, what is the temperature and oxygen at a half a meter off the bottom, at 1 meter, at 2 meters off the bottom, and I can plot that. Then what I can do is go through, just clip that little zone, that selection curve out, scale it to 1, and I've moved beyond temperature and oxygen. Now I have a probability of occurrence for walleye at the different depths. When I create those maps of where those nets would sit, we can see here's gillnet 7 on Gull Lake. We can keep track by mesh the depths of where they go. That first 50 feet is sampling kind of that zone of, looks like 4 to 6 feet. Pretty good walleye probability. All the way down to 20 feet. It's a pretty good walleye net. Contrast that with this one that's kind of set in this offshore saddle here, that these 2 center mesh, actually these 4 meshes are all in zones that we wouldn't expect there to be very many walleye there. So if we look at the seasonal, that second week of July when we set this net, certain depths might not be very good. If we were looking at between year comparisons, maybe that's just because it was a little bit warmer that year or the water was a little bit more hypoxic. So then what we can do is apply the standard method of when we always do these profiles to calculate the probability of where they might be. So putting this all together, we have a relative hypsographic curve here. 100% of the water is at 1 foot down as we go through. I can apply that model to the depth. We don't have some nulls down here at the bottom. And I can overlay where those nets sit. And so we get a pretty good idea of maybe we wouldn't expect there to be very many walleye in those lowest catch rate nets. So another piece of the story here is that's just for walleye. I've subsequently done the one for Cisco. But another tool that we can use, I flipped this hypsographic curve around so it's kind of like looking at depth right here. And here's 10,000 acres, the shallowest. This is kind of like the bottom contour of a lake. I know where my nets fish. Relative to that bottom contour, I can say, well, here are the areas of where they are. So Cisco are that offshore pelagic fish. They're moving out here in this open water. And we can use hydroacoustics to generate distributions where they are. And we look here, I just scaled everything to 1 to show during the same time as our gillnetting, well, where would we find fish? Well, this is the actual true depths of where they would be with the relative densities from 0 to the highest point here. And very unlikely would we encounter a whole lot of Cisco. So, and I'm going to come back to why that's important in a second. So why is all this important? take a look at our standardized assessment techniques. We've set our monitoring programs up to look at these fixed time, certain calendar day of the year, and depth locations. But if this habitat's changing, the species that are associated with that unique habitat, or maybe they're just moving to different areas. Are we monitoring what's going on with the population, which we're hoping to do, or are we just monitoring what's going on in that type of habitat? We can look at these unique species assemblages, looking at what's going on with Cisco, and again, changing habitat conditions, looking at climatic patterns and increasing the numbers of nutrients. I'm going to end moving into this efficient conservation, and this is what Pete would have come over and talked about. Here's his title slide right here. Cisco are important cold water fish with catch rates that are declining, and here's that same declining trend. And is this real? We have these die-offs that go through. Important forage species we want to know as managers of where they are, or is it an artifact of maybe some of these other things? So Pete has really taken the relationship to the lakes where they are and are not is certainly a real thing. We're starting to see populations in some lakes, especially in the southern, decline, but in real when we've gone out with hydroacoustics, not just from our standard netting, from targeted netting. So what Pete did a few years ago is they did some modeling down with folks at the U of M to be able to say, given these levels of climate change, how would we identify Cisco refuge lakes? So we're not going to be able to do anything about how much temperature is accumulated. We can't put chillers out in the lakes. But what we can do is limit the amount of nutrients that go in the system and use up that hypoxic water. And what they were able to do is, given any level of, or given some of the most extreme levels, we'll still have of that 650 lakes, 176 lakes that are deep cool water refuges. Then what we want to be able to do is prioritize, maintaining these, and maybe some of those middle, improving them to come through. So the key of what they found looking at some of these relationships of the densities of fish and the land use practices is we really need to have a good surrogate for the health of that watershed is the amount of forest that's in that watershed. And it's been a fairly unique thing. In the past few years, we've had this Clean Water Land Legacy Amendment go through that was a self-imposed tax on Minnesota residents to go through. That's a very small percentage, but it's going towards 3 or 4 elements, but one of them is protecting clean water. They've been able to go in and use some of these funds to buy up, especially in the case of kind of the 10-mile lake watershed, to be able to make sure we're at that 60%, but help prioritize. And to date, they've spent $4 to $5 million putting easements on land to help make sure that we can protect this important this hypolimnitic oxygen will be an important or increasingly valuable ecological resource. Then when we have this money specifically dedicated to how we might conserve and preserve some of that habitat, how do we effectively do that conservation? So Pete's been able to go through, identify these landscape parcels to try to make sure that those ones that have been identified as good refuge lakes, we can continue to keep at those levels. And we don't want to be putting money into systems that we know, we'll never be able to protect them given those levels coming. So just strategically use these really large amounts of money that usually we don't have. So Pete's been working at this for probably the past 5 years, but developed some great kind of landscape tools that allow us to effectively administer and put these monies in places, working with partners, counties, soil, water, county district, to be able to go through and preserve that land. So, with that, any questions?
The state of Minnesota has planted spotted muskies or Great Lake muskies. Wisconsin has decided not to do that. Is there a correlation between the cold water Cisco populations and influencing those decisions? Do you know?
For stocking muskie, I can speak to Minnesota practices, the leech fish I assume we're talking about, maintaining the right genetics that will match up with the species that will do well in those conditions. And we try to match up. Part of our stocking plan relies heavily on trying to make sure that we don't move fish between basins that will match up. So the fish populations that are in that Leech Lake, or the Mississippi strain is what we call them, all part of Leech Lake there, to be able to match up with those forage that is found native that they would have been found with. And that's one of the practices that we adhere to try to make sure that we're matching up the species that has evolved in these habitat areas to the natural forage that might be in there. So, and I know as an overarching principle, maintaining those native genetics, I mean, they often will do better in those systems, but that's one of the kind of the bigger picture principles of, in some cases there can be growth rates. We've had shoepack ones, or the ones that we had a learning curve in Minnesota of how to deal with. These were some that were up in the northern part of the state, a small lake, and there was some kind of genetic component to it that they wouldn't attain large sizes. Part of it was environmental, but the other side of it is that they truly just couldn't get those actual sizes. And we stocked those fish all over the state, all over Minnesota in the 70s and 80s and even into the 90s. And then in the late 90s, we really started taking a little bit closer look at that to try to make sure. And it's really interesting to see that even despite all of those stocking efforts, the local fish in those systems, if they are still there. So in some cases, we've actually gone in, we've genetically marked some of our fish and are able to remove those high percentage shoepack genetics out of these systems to keep those ones from kind of cross-contaminating with those local strains that are there. So within Minnesota,
if Cisco populations drop, do you have any data to say here's the forage fish that the walleyes, northerns, muskies might be going after? Panfish?
Well, they certainly can, and they do as they go through. It's those other categories. And certainly we have muskies and walleye in lakes without Cisco as a forage. But we'll start looking at those trade-offs in growth. So when you have this predator or forage that's relatively easy to catch out in these open water, you think of a big school, especially if they're squeezed down into this little zone that's 2 or 3 feet tall, you have a muskie going out there, it could probably close its eyes and swim with its mouth open and encounter these fish to go through. So without that really, I mean, otherwise if they're up near shore chasing perch through vegetation, I mean, just energetically they don't get the bang for the buck as they go through. So that'd translate out to how long does it take us to grow these big fish. And we know that they can eat suckers, other kind of soft-rayed fish are kind of the preferred as they go through. But if they do have, it appears to be a pretty good strategy because they can grow quicker on these systems that do have Cisco. And that's one of the things that our managers want to be in front of to be able to, I mean, if we do deal with systems where we don't have this energetic, would we maybe have a regulation because then growth might slow down for these fish? Would you maybe have a subsequent generation that could offset that to continue to provide the fishery or the similar type of fishery?
Are these data comparable to places in northern Wisconsin in terms of like dissolved oxygen and temperature?
Yeah, I mean, there's I think as we look at the kind of the latitudes as we go through, a lot of it has to do with the refuge of how deep your lakes are. So further south in the state, I'm sure you have some deeper water lakes that'll have that cool, oxygenated water that these fish could do well. I mean, if we kind of shift a lot of our lakes, or maybe 40% further north than yours, so the further you go north, the more kind of cool water habitat you'll get to go through. But I think the same principles would probably apply for these selection. And I mean, as we look at kind of the cool water, we're just a little bit further down, further south away from that. So on average, all other things being equal, the lakes would be a little bit warmer, but you'll always have those exceptions to go through. And that's what some of Pete's work could help identify. If there are these lakes that have, I mean, a good cold water, no matter how much we warm up that upper water column, well, if we limit the amount of nutrients or even scale back, we might be able to preserve those. So even in the southern part of the state, we have some of those lakes, as I'm sure you guys would as well.
So say on a lake that has both forage of yellow perch and Cisco, for example, Lake Vermilion, right? With walleye, how many fish do you guys have any data that you found showing the size distribution of fish that are chasing the perch versus chasing the Cisco?
I mean, from an it's really hard to tease that out. From a big picture perspective, we'd go back. I mean, how much effort would it take to catch a little perch in vegetation near shore. And then what would be the reward on that? And we can look at how that trade-off and then plays out with, well, how big do these fish get? I mean, you look at mille lacs. Mille lacs is an example that we've grown some of the biggest muskies we've ever seen out there. And angler catching dynamics has changed, but there's Cisco, even despite some of our modeling. If you look at these temperature oxygen selection, we'd say Cisco shouldn't be in the lake. They're still there. They grow really quickly and die pretty early on, but provide an incredible resource for these fish. And if you'd look at how long it would take these muskies to attain those large sizes, spending that time chasing perch around, for sure, sometimes of the year they would. If there weren't Cisco in the system, they would spend that time doing. But when we look at that, I think it'd be similar to some of the work that Brian's done. I mean, eating fish that are a larger size. I mean, if you're a 40 or 50-inch fish, you need to be eating fish that are a foot long or so, really, to get those trade-offs. You can't kind of make a living eating 4-inch fish. So.
Can you comment on the introduction of zebra mussels to any specific lake? Does they create water clarity?
They do.
Can you just comment on that? You know, how big a problem is that?
Well, I think problem lies in how you might define it, where it goes through. It's going to change the system ecologically. So when you clear up a lake through filtering all this through, well, we're going to get light deeper in lakes, which is going to cause plants to grow deeper in lakes. When you have this changing, and that has really interesting implications, you think where we set our nets. So maybe in the past we set it on that deep side of the weed edge. Our lakes, most of our lakes now have zebra mussels, or within 5 years they'll have it. We'll see the implications of that in another 5 years to go through with increased water clarity levels. So those fixed sites at depths are no longer sampling the edge of the weed line, they're right in that vegetation. So what does it mean? It changes the way the productivity is divvied up in the system. At that primary level, they're removing a lot of those nutrients. That could have gone towards larval fish dynamics. There's all different ways it could have gone. So as managers, sometimes it can kind of skip a level in the sport fish of interest. That's why I kind of am hesitant on problem. It might benefit some species, but not others. It changes the way our anglers have to fish in lakes, which is a real perception that we would see. But maybe the bites are just concentrated during the day or daybreak and sundown. So, but it's one more critter in the environment that's taking that limited piece of productivity pie that you have, and that's going to translate through all the other elements kind of in that food chain. So,
Thank you.
Mm-hmm.
All right, I think we probably better vacate the premises. Thanks, everybody.
Yep.